In this video, we're going to take a look at some of the material from Chapter 10 in our OpenStax textbooks. And in the first part, we'll take a look at some of the experiments that are used to identify the factor that was actually responsible for heredity. And we're going to look at how people use some of the microbes out there to do this process. And in the second part, we'll take a better look at the structure of the nucleic acids and also the genome. If we go back to the early 1900s, we can take a look at the work of a guy who was working with a single-celled algae, and his name was Hammerling. And what he was doing that was really kind of interesting is he found that he could take these cells, and you can see from the picture over here that these are actually pretty big single cells here, but he was able to go in and physically dissect and remove parts of the algae that was out there, and then he was able to take these and make some observations on whether or not they could actually regenerate the parts that were removed. And if you remove the top part of the algae there, he found that a new cap could actually regenerate, as we can see in this top part of this figure. But if he removed the bottom part, or the foot right here, he found that the foot would actually not regenerate as a result of that experiment. So again, this is, if you look at the scientific method, this is an observation that leads you to scratch your head and to start coming up with a hypothesis, a proposed explanation, and then use some experiments to try and prove the truth or that your hypothesis is not correct. And what he did is he went on to look at some more of these algae out there, but he did something that was kind of interesting. He was able to take a different species of the algae out there and graft some pieces onto the different types of algae here. And so if you look on this thing on the right, you can see these two different related species of algae out here do have some differences in the part of the algae that could be regenerated. And so he was able to take and basically chop out a middle portion of the algae and replace it with something else and get that graft to be functional. And then he would take a look and see what was actually happening as a result of that in terms of the new parts of the algae cell that could grow out. And he found that it didn't matter what the middle portion of this graft was, but really the important thing was wherever the foot was, that seemed to direct the structure of the new parts to the algae cell that were being made there. And so he concluded based on this that the information needed to regenerate that top part there must have been located in the foot. And conveniently enough, we know that that is where the nucleus is in these cells. It's actually in the foot. So it appeared that the information that was necessary to regenerate this top part must be stored in this lower part right here. And it was passing through that graft, and that the directions were not stored in that grafted on piece there. If we go up a few decades and hit the 1940s, we can see another experiment that was done with some microbes. And in this case, Beadle and Tatum were actually looking at the ability of some different types of molds to grow. And what they found is, based on some previous work, X-rays actually resulted in mutations in different organisms. And by using the molds, they could actually grow these things up in the lab and expose them to some X-rays so they could get some mutants that would form there. And what they did that's really kind of interesting is they took these mutant spores, and remember, when we're looking at the fungi that are out there, they're haploid, so they only have a single copy of their chromosomes, and mutations that would appear in this could potentially result in lethal phenotypes where you would not see those. But what they did that's really kind of a unique thing is they were able to take these and cross them with some wild type spores and grow them out, and they would grow them on complete media. So think like nutrient agar. It's got all the nutrients that these organisms need to grow, and then they could take that and grow them on a minimal defined media that was lacking a lot of the nutrients that were available in that complete media. And if they found things that could not grow on the minimal media, they knew that something must have been going on that disrupted certain pathways that would be involved in making the food, or really taking the nutrients and processing them to allow these molds to grow. And so they took these ones that could grow on the complete media with everything they needed, but could not grow on the minimal media, and then they went through the laborious process of testing these by growing them on some minimal defined media, where they supplemented it, in this case with single amino acids that were purified there. And what they sort of came to the conclusion on this is that spores that could grow in these tubes must have a mutation in a pathway that was producing that particular amino acid. They could not make that amino acid for themselves, but by adding it, they were able to get these organisms to grow, and they could grow on the complete media because that did contain those amino acids already, and these molds did not need to have that amino acid supplemented like we see on our minimal media there. And when you take a look at what's going on here with their mutations in the spores that could actually grow out on their complete media, when they tested them, they found that there were 3 different classes of mutations for the arginine mutants that were out there. The wild type ones, the normal kind that you would find growing out there, those could grow on anything for their minimal media. And they could supplement it with these different amino acids or different intermediates that were involved in the pathway for the synthesis of this amino acid arginine. And if you think about this, each of these classes can grow on different types of minimal media that contain these intermediates right here. And so if you think, these guys did not grow because they were not able to make any of these kinds of things that were out there. These guys were able to grow when you added this ornithine, so that maybe they were able to take that ornithine and process it into citrulline, and then process that into the final amino acid there for arginine. And what they concluded as a result of this is what's known as the one gene, one enzyme hypothesis. And if you think about this process here, there must have been a mutation in a gene involved in the arginine biosynthesis process. And if that mutation was in this first enzyme here, they couldn't make any of these products here. If the mutation was in the second enzyme right here, if you supplemented them with ornithine, they were not able to process it and make citrulline or the arginine. And then the third one over here, a mutation in this third enzyme, basically blocked this last step in the arginine biosynthesis pathway. So their conclusion is that each gene was actually encoding a single enzyme that was involved in this pathway here, and that they had made mutations in those genes totally by random chance with their X-rays when they exposed the spores to that. If we back up a little bit and go all the way back to the 1920s, we find a really interesting set of experiments going on to try and identify the molecule responsible for traits. And this was done by Fredrick Griffiths' lab at the University of Chicago, and he was looking at 2 different strains of Streptococcus pneumoniae that were out there. And if you look at this figure I got from the web out there, their phenotype of these 2 strains was different. Everything else, when you tested their biochemistry, was similar. When you did staining, the only difference that you could see is the colonies that were rough like this actually were lacking in capsules, but these smooth strain over here, these guys do have capsules, and you get that almost wet appearance to your colonies. The other difference is when you infect mice with the rough strain, they actually were able to survive a lot of that and did not develop pneumonia like what you would find with the smooth strain. And so this strain over here, the capsule, was actually able to protect the cells so that the immune system in your test animal could not clear the infection, and they would develop pneumonia as a result of that and die off. And so Griffith was looking at some of these properties of these 2 different strains of the pneumonia bacteria, and he was testing them out. And if we look over here, you can see your control experiment with your rough strain. These guys are non-virulent. The mouse was able to live. If you took the smooth strain and injected it into the mice, they would die. So there is that distinct difference between the 2 different strains. But then he started messing around and killing off some of these smooth strain cells with heat and injecting them into the mice to see if it could still cause death of the mice. And if you see from the results here, the mouse actually was able to live. But when he took live rough strain cells and heat-killed smooth strain, and if you think about this, both of these tests resulted in the mouse living, but when he mixed these two together, all of a sudden the mice were dying. And when they went in there and recovered some of the bacteria from the bloodstream of the mice here, they found that those live rough strain bacteria had somehow been changed into live smooth cells that were out there. And so what he concluded is that there must have been something, what he called the transforming factor, that came from those dead smooth cells that was actually able to change the live rough cells. And this is a really startling demonstration here, because this is really the first example where he was able to show that the transmission of these traits did not have to occur just between generations of cells. This was actually a transfer of traits between cells of the same generation. It wasn't until about 20 years later that we find another lab was able to do further work on Griffiths' transformation experiment, and that was done by Avery's lab in the 1940s. And what they did is, instead of using the cells, they found they could actually make an extract from those cells. And so imagine taking your culture of cells and throwing them in a blender and breaking them up into little pieces. And then they could take that smooth cell extract and use that to actually transform the rough cells, so that when you put them onto some plates, you would get your smooth cells coming out of there. So your smooth cells were dead, but they were still able to have that transforming factor. And what they did is, when you have an extract from cells, you can start to basically dissect it. And in this case, they used enzymes to break down the different kinds of organic polymers that you would find in those extracts from the cells. And so if they treated them with these different kinds of proteases or ribonucleases, those would break down the polymers that were in those extracts. And they found that when they did this, they were not able to disrupt the transformation. So we know from this that proteins and RNA probably are not the transforming factor. But when they treated their extract with the enzyme DNase that degrades your DNA, that one seemed to actually block the ability of that transforming factor to work. The problem with this is it's negative data that you're looking at. We were able to degrade something and it resulted in a lack of something happening. And people said there is a possibility that you might have some protein contamination in your extracts, because there is still a lot of debate on whether or not the transforming factor, or the factor responsible for heredity, was DNA or proteins. And a lot of people thought it must be proteins, because they are made of all these different kinds of amino acids, and they couldn't imagine that you could have this variety of traits just based off of something that contained 4 nucleotides in it for your DNA. But really the definitive proof came around with an experiment that was done with some bacteriophages in the 1950s by Hershey and Chase. And they were able to grow up their phages in a culture medium containing some radioactive isotopes. And so they were able to label their phages, either the DNA with some radioactive phosphorus, or they could label that protein coat with some sulfur, because DNA does not contain any sulfur here. It's only found in the amino acids that are out there. And so they could grow up these phages and label them with these radioactive isotopes, and then track where the DNA was going and where the protein was going. And so they went ahead and did their infections, and they did not know this at the time, but we know it now, is that phage would attach to the cells and inject its nucleic acid into the cells there. And then they took them and put them into a blender again to break it up, and then centrifuged it, where you take that extract that's left over there and you spin it in some tubes at really high G-force to separate the components. And the cells in this case are going to sink to the bottom, and your liberated phages are going to remain suspended in the liquid up there. And what they found as a result of this is that the cells were able to actually sink to the bottom, and that's where the DNA was going, but the protein remained floating around in the solution up here, what's known as the supernatant. So they concluded from this that the phage DNA is what stayed in the cells, and if it was injected into the cells, that must be the factor that was responsible for the heredity. In this case, it coded for making new viruses that are out there. And you can see from this last figure here that there are a variety of events that led to the identification of DNA as the molecule responsible for heredity. And we're talking about something from a 100-year time span here.